Awesome. So in this presentation, I'm just going to give you a little overview of 6 tasks that we use in our Year 10 genetics unit to help develop students' genetic literacy. So to that end, I thought we'd start by looking at what genetic literacy actually is. So, a one-sentence definition of genetic literacy would be something along the lines of the knowledge of genetics that is relevant and useful for the non-scientist citizen to possess, and which they need in their everyday lives to make well-informed decisions and to develop well-informed views as well on some of those social genetics issues. To delve a little bit deeper in terms of what knowledge is actually needed for someone to be genetically literate, the best place to go for that, in my opinion, is a study done in 2017 by some researchers in the Netherlands. What they did is they sent out that question: what knowledge of genetics do non-scientist citizens need in the 21st century, to about 53 experts in the field. They fed some ideas back. They then brought those 53 experts to Utrecht for a conference, and they asked those experts to examine 7 case studies involving real-life scenarios, things like buying a direct-to-consumer genetic test, interpreting a news article regarding a gene for alcoholism, and buying genetically modified food. And they asked them just to check that the knowledge ideas that they had sent in fully covered what a citizen would need to know in each of those scenarios. The product of all of that work were these 3 tables: 9 what we call conceptual knowledge components, things like, all organisms have genetic information in their DNA, proteins are central in the structure and function of living organisms, and then number 9 down there, multiple genes and multiple environmental factors interact in the development of most traits. And that 9th conceptual knowledge component down there was the one that most of the participants in the study said was the core message of genetic literacy. We then have 2 other sets of knowledge components: the socio-cultural knowledge component and the epistemic knowledge component, which are equally relevant in terms of developing our genetically literate citizens. So in this, I'm briefly going to talk you through 6 tasks that we do that address various aspects of these ideas regarding genetic literacy. Our genetics unit at Year 10 is based on those 3 tables that you just saw, and so we try at various points throughout the unit to address each of those knowledge components. And as I said, they're treated like a smorgasbord. You might like none of them, you might like one. Take from it what you will. So the first task that I was going to introduce was just one on proteins. And as that 3rd knowledge component pointed out, proteins have a central role in the structure and function of all living organisms, and we think it's really important that students understand that. This is a reasonably straightforward task. The students get the DNA sequence of 10 genes. They work out what amino acids the protein is made of using a codon wheel, and then they go around the classroom and read the profiles of different proteins and work out which protein each gene codes for, and then write a little description of its function. So it's quite a straightforward task, but the nice thing about this task is it introduces them to a wide variety of proteins across various different organisms. One thing that's very clear from the research, and there's been a few groups in the US particularly who have looked at teaching molecular genetics to Year 7s, 8s, 9s and 10s, and they all come away with the same conclusion. They wish they had spent more time actually talking about what proteins do, and not just 1 or 2 proteins, but many proteins, so that students can truly understand that proteins are the workhorses of organisms. Everything that happens in an organism happens because a protein somewhere is doing something. And so this is a task, one of several tasks we use to try and get that message across, and I've provided the link for it there. Okay, the second task is a task that focuses on kind of the basic mechanisms of inheritance. So it's a task that focuses on how sperm and egg come together to produce an offspring, and how the sperm carries one allele and the egg carries another allele. And we draw a Punnett square to show the possible genotype combinations. What's different about this task is the context. So rather than the outcome of this task being a student saying, for example, there is a 25% chance that the offspring will develop Huntington's disease, or there is a 75% chance that the offspring won't develop cystic fibrosis, The outcome of this task in terms of the kind of statements students will be making are more probability based. So for example, there is a 25% chance that the child will have an 80% probability of thinking coriander tastes like soap. So the context for this task is different, in that the context for this task is a multifactorial trait affected by multiple genes and multiple environmental factors. And so we introduce the idea of inheritance through a trait like this in order to make sure that students recognize that multifactorial traits are the norm, and they should be at the core of their thinking when considering genetics. And I provided the link to this task here. Okay, the third task is actually a task developed by the National Institute of Health in America, and it focuses on understanding how height is a multifactorial trait, and it's actually a modeling task. So it's a physical interactive task where the students will model the inheritance of height. In this particular task, height is positioned as the product of 6 genes and 3 different environmental contexts. And students use beads to determine which alleles are inherited for each gene and which environmental contexts are present for each individual at each stage of development. Each combination of alleles and each environmental context is associated with a particular numerical impact on height. So if they have 2 red alleles for gene 1, then that might be plus 5 centimeters. If they have the orange bead for the prenatal period in that child's life, it might mean that the mother consumed alcohol during the pregnancy, and that has a minus 10 centimeter effect on height. So it's things like that. Those all then get summed together to get a final height. Now, if every student in the class does that, then what you can get actually is you can model the inheritance of almost a generation of 25 or 50 people if you do it for female and male. And you can plot all of the heights generated by students on a histogram to show that the kind of continuous variation that you would expect with a multifactorial trait. And so it's a good way of kind of showing that if a continuous trait like height is best explained in the kind of multifactorial, with the kind of multifactorial model that this is. You can also do this task as with positioning height as a monogenic trait, where you just have one gene, a couple of beads, every student in the class picks their beads out, and you get a height out of that. And then you can compare the degree of variation that you get in your, I suppose, offspring generation when everybody in the class is modeling height as a monogenic trait versus everybody in the everyone in the class modeling height as a polygenic trait. The nice thing about this task is that it has a low kind of there's not an enormous quantity of knowledge necessarily that students need to carry out this task. We don't talk about what any of the genes do. All of the environmental factors are things the students will quite readily understand or know of to some degree. So it's quite a good task just to encourage an appreciation within students for the kind of multifactorial nature of traits like height. Okay. On the other hand, definitely a slightly more difficult task is a task that we've got, and you go to the link, you can get the full task with all of the images in the talk through. This is a task focusing on developing students' understanding of eye color as a trait affected by multiple genes. And we decided to do a task on eye color, one because students are naturally very, very interested in eye color, but also any textbook you go to almost you can find people talking about alleles for blue eye and alleles for brown eye, and we really wanted to get students away from that idea and open up a little bit of the kind of black box that defines the genotype-phenotype relationship when it comes to eye color. So I'll briefly talk you through the knowledge that we impart to students in this task, and then the full task you can have a read through online by clicking that link. Effectively, we teach students that eye color is determined by what's going on in your iris cells in your eye, and we tell them that in the iris cell there is a structure called the melanosome, where the melanin is made. And when we teach students that the more melanosomes there are in an iris cell and the more melanin gets made, the darker the eye color. We then introduce them to 2 proteins, tyrosinase, which is an enzyme, and something called the P protein. And the tyrosinase enzyme, they learn, is responsible for producing melanin, and it sits inside the melanosome, and the P protein builds the melanosome. We then tell students that there are 2 alleles. So we are simplifying a bit here, obviously. We then tell students that there are 2 alleles for each of the genes that code for tyrosinase and P protein. One that codes for a functional tyrosinase, one that codes for a poorly functional tyrosinase, one that codes for a functional P protein, and one that codes for a poorly functional P protein. We then give the students 4 or 5 genotypes. So you can see down there we said John's iris cells contain the genotypes TP and PW. So the TP would be the poorly functional version of the tyrosinase allele, and the PW would be the well-functioning or the good-functioning version of the P protein allele. And then we give the students a bit of time. We give them time to discuss with their peers and discuss as a class what they think the phenotypes will be for those people. And we ask them to comment both on the phenotype of the individual person, but then comparatively, which is actually an easier judgment call to make. And even though the knowledge threshold here is quite high, I have found that the students, with a bit of time, are more than capable of coming up with some predictions as to whether they have dark brown or hazel or blue eye colour. For our students, we don't ask them to memorize this. The goal of this task is just to develop an appreciation for the idea that eye color is polygenic. We don't expect them to go away and memorize details about tyrosinase and the P protein. So I suppose the goal of the task really is one of just having an appreciation for this idea of complexity that feeds into eye color. But yeah, even though this is a task that requires a bit more knowledge, I have found that students are normally more than capable of rising to the occasion of deducing a prediction from the genotype. Okay, the 5th task focuses on monogenic traits, and the link to the full task is there. It's a worksheet that runs through a series of activities, and the goal of this task is really about getting away from some of the misconceptions about dominance that exist amongst students. And probably there's nothing in genetics which causes more misconceptions than dominance and recessiveness. One of the big misconceptions, of course, being that this idea that we have a dominant gene and that suppresses the recessive gene, and the kind of nonsense like that. And so to counter those kind of ideas, we focused, once again, came back to the bread and butter and tried to recenter the discussion around proteins. And so what we do in this task is we introduce students to cystic fibrosis first, talk about what cystic fibrosis is, and we talk about, obviously, the CFTR gene as being coding for a protein which pumps chloride ions out of the cell. We then draw for them, as you can see on this diagram on the right, what the phenotype of a cell would look like if it had one normal allele that codes for functional chloride ion transporters and one mutated allele. And so they can see that even though there is a mutated allele in there, because there is a normal allele, the chloride ion pump still gets built. We then get them to draw what it would look like if there were 2 mutated alleles and 2 normal alleles as well. The nice thing about this is that we then compare it to Huntington's disease. And obviously the nice thing about comparing Huntington's disease and cystic fibrosis is a recessive condition and Huntington's disease is a dominant condition. And we talk to students about the fact that the gene that for that Huntington protein, the dominant allele that causes Huntington's disease, does so because it codes for a toxic protein which forms aggregate clumps, which can then block neurons and stop them from functioning. And so that the nice side-by-side of that is it allows students to understand that the recessiveness and the dominance of those 2 illnesses stems from what is happening with the proteins that those genes code for, and nothing else that they themselves decide that's how dominance or recessiveness works. And it's nice for them to think about, okay, well, in Huntington's disease, it's a dominant condition because you only need one gene, one allele, to produce the toxic protein that will destroy the cell, so to speak. Whereas in cystic fibrosis, you would need both alleles to be mutated for you to have no chloride ion pump capabilities. And so this is a task which focuses very much so around getting them to draw what they think the phenotype looks like, and so they can understand it at a protein-based level. The link to that is there. The final task is just one that focuses on genetic testing. We get the students to look at a sample 23andMe report for a person who has been tested for one of the genes involved in Alzheimer's disease development. We have a discussion with them about what Alzheimer's disease is and the various factors that affect Alzheimer's disease, and then we get them to do a worksheet basically that runs through a series of questions on that particular topic. It gives us an opportunity to discuss some of the personal implications, I suppose, of genetics. Okay, so that's very much a whistle-stop tour of 6 tasks that we do. If you go to that link, it's a long page of lots of resources that we use, but they're organised by topic, so they should be easy to find. If you've got any questions or if you're interested in more information, feel free to email me. But hopefully there's something in there that's useful.